Thank you so much. You're my hero, always.
And you're my Superman, so we are open now.
Okay, thank you. So we'll give a couple of minutes because people, have lots of other things. So we'll wait. We could still chat. So how about the climate problem in your country?
I actually have also included the slide. I'm 20 kilometers away from a brown coal open pit mine, and we are getting a new government just in these weeks. And we had plans to get out of coal, although it's 40% of our electricity, 45% by 2038. Now the new government says in 2030, so in 9 years, we want to be out of coal, although it's more than 40% of our electricity. Oh. And so in Germany, bioeconomy funding is really, it's a great time to be in synthetic biology, trying to do something for the bioeconomy, sustainability. It's a great time because there's so much pressure.
I see. Actually, I've been in Germany once when I was in company. Actually, I visited Stuttgart at the time to buy rheometer. And as I said, I was working on the biopolymer at the time. So we bought the rheometer, we got trained over there, and then I visited some beautiful place near the Stuttgart. And then I still remember that, but that's a long time ago, almost 20 years ago when I was in.
My family comes from Essling area. That's the next town from Stuttgart.
Oh, really? Interesting.
I was raised in the north. Yeah.
Okay, I love the place, and I tasted the most delicious beer in the Stuttgart with the sparkling beer, and then I never tasted that one. I never found that beer again, and after I just left the Germany at that time, I still miss that one. So Tal, how in New York right now? Actually, I watched the NBC Christmas tree kind of event yesterday on TV. Lots of singers over there, and that's got tradition, I believe, in New York City, right?
Yeah, Thanksgiving Day parade and Christmas tree lighting. Very nice here around the holidays.
Wonderful. So let me see. Okay, so I will give up. Okay, okay. So let's start. I mean, so we now my time 10:03. Okay, so good morning, afternoon, evening. Hola, hello, bonjour, ni hao, konnichiwa, annyeonghaseyo, and I think guten tag. And hello all. Welcome and thank you for joining us. The 12th seminar in Germany SynBio Day. We had 2-week break due to Thanksgiving holiday. It was so great for me to catch up with friends over dinner and finish proposal reviews for DOE and NSF, as well as review multiple papers for PNAS, Nature Journal, etc. And I hope my reviews are more generous due to my relaxed mood at the time. And also, I am so thankful during the Thanksgiving holiday. This seminar series is indeed at the steady state now. And I found the 11 seminar videos have attracted, as of now, total more than 10,000 views from roughly 1,000 global audience. This is all thanks to young rising star and pioneer speakers, as well as the excited global audience. So I'm truly thankful for your attention, time, and enthusiasm from Africa, Asia, Australia, Europe and America, as well as potentially internet-savvy penguins. And despite all sad news regarding inequality, climate crisis, unreasonable shooting and killing, and COVID, I strongly believe we can change this world together by giving our time, resources, attention, support, compassion, kind words, empathy, forgiveness, and of course, our love. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Lars Blank. He is a professor at RWTH Aachen University, Germany. I personally like him a lot because we share many things. He is an associate editor of Metabolic Engineering Communications, where I also serve as an editorial advisory board member. And one of his early papers on microbial hyaluronic acid production is my favorite because I produced this biopolymer at a commercial scale using 1,500-liter bioreactor, and we also got FDA approval when I was working in the industry. More importantly, he is an expert in plastic upcycling, which is one of my enthusiastic research topics. Professor Blank, the virtual floor is blank now for you to fill in.
Yeah. Professor Moon, thanks for the extremely kind introduction, and thank you for the invitation. And I'm really happy to address the young scientists, and I'm really looking forward to Tal's seminar. So just sharing a few thoughts. I put it in my Excel sheet that it's really an interesting time in Germany, where we located 2 kilometers away from the Dutch border, 3 kilometers from the Belgian border. And it's a very interesting city, but also importantly here, we are 20 kilometers away from a coal power plant area, open pit mining, and our new government will drop out of coal in the next 9 years. And there's a lot of money now flowing in that we come into a bioeconomy. So synthetic biology will have ample opportunities, and you see the impact the current linear economy has on the environment, besides obviously the CO2 we all feature. And we had these floodings, and I will address it also, but I would like to emphasize that if you're early in your career or just thinking about something, you might look at the really large problems, and I think these world limits is a good idea to look at. Climate change, these scientists said we are still in an area where we can change something, but nitrogen and phosphorus are even worse. They are out of limits, out of bounds, and you may want to have a look also what you can do on phosphorus. We are looking into that as well. But today I just want to share 2-3 minutes on carbon, and just one aspect, Professor Moon mentioned it already, and this is plastic. We learned as humanity that this is a very nice material. Crude oil was a very cheap substance, and since then Asia kicked in, and we kept developing, and we are getting close to 400 million tons per year newly produced plastics. And if you look how this is done, it's all the time moving more and more to Asia, while the US, NAFTA, Europe is maybe even going down, because it's so a commodity these days. And we produced already as humanity 8,300 million tons and recycled less than 10%. And here, this is an ideal case. We have the plastic in one spot, we may incinerate it or we may recycle it, but you saw these pictures, you may have seen it depending where you live in your community. We have a problem here. And this is one of our partners. This is not anywhere, this is in Europe. And they developed a small ship that they can remove from waterways again the plastic. And if you're interested in how this is possible, you may want to follow our project in this blog. Here you see a different aspect of all these topics. We had huge flooding, unseen floodings in Germany. And now we have all the debris in our waterways, and they came back to Germany and actually trying to fish out some of the mechanical stuff. Yeah, what are we trying to do? We are, I'm coordinating a joint effort between Europe and China, because China is one of the 30% of the plastic produced. Europe, 16% of the plastic produced, half of the plastic in these two continents. And we are trying to use microbes. And these are enzymes degrading plastics. And these monomer mixtures, we are training microbes that they can utilize this as carbon source and then produce PHA or other products of value. And a lot of synthetic biology from the enzyme production to the redesigning metabolic networks and coming up with interesting microbes which produce products of value. And this may be a vision where we use the bow tie structure of metabolism to fuel the different monomers and produce products of value. And here I hope I put in hyaluronic acid. No, I did not. I did not put it in. And here just some results. We can use now a lot of monomers, also joint monomers, to produce PHA and biomass. And yeah, this is from our partners. They produce truly PHA in resin to make real products out of it. And we put it into a vision. Obviously, we have to think about reuse. Mechanical recycling for PET, for example, is now giving really speed. But here we are also contributing in a recycling, and we have to change. And there's so much opportunity for synthetic biology. The carbon source from crude oil, we have to move to biomass, CO2, and obviously the recycling stream. And we don't do full recycling. It will be open recycling, so combustion is still in there. And I could talk longer here, but it's Tal's time. And so what do we have to do? Everyone can do rethink, refuse, reduce. And at the end, if nothing left, we recycle and then we go on. So if you're new to synthetic biology, if you have your career in front of you, take the big challenges, and synthetic biology will contribute. And thanks again, Professor Moon, and thanks, Tal. And I'm stopping here.
Wow, thank you so much. I mean, thank you so much. Wow, that's brilliant talk and inspiring insight. And I completely agree with you. This is the very important problem. I would say now climate change is no longer the right term. Climate crisis or even emergency should be the right term for current situation. And then plastic is one of the main contributor to that, critical problem we currently is facing, and then we need to solve that problem as soon as possible. Otherwise, there is no future for my daughter and my younger generation. So thank you so much. I mean, that is absolutely wonderful presentation. Okay, so now the main speaker of today with a longer introduction. So Tal Danino is an associate professor actually in the Department of Biomedical Engineering at Columbia University. His lab focuses on engineering bacteria for biomedical applications with a particular emphasis on developing bacteria as a cancer therapy. Originally from Los Angeles, Tal received a PhD in bioengineering from UCSD in Jeff Hastie lab and was a postdoctoral fellow at the Koch Institute for Integrative Cancer Research with Sangeeta Bhatia. He is also a recipient of numerous awards, including the NSF Career Award, Era of Hope Scholar Award, CRI Lloyd J. Old Stars Award, Pershing Scared Son Prize, and is also a TED Fellow. To just name of several. He directs the Synthetic Biological Systems Laboratory and is also member of the Harvard Irving Comprehensive Cancer Center and Data Science Institute. Personally, my lab and I love his work since he was a graduate student at Jeff's lab, and he and I almost became an academic brother when he was considering joining the Chris Voigt lab at UCSF. I also thought, given his great achievement in probiotic engineering for cancer therapy, he is a tenured professor who is not eligible for this seminar series as a young speaker. When I visited Columbia University 2 weeks ago to give a talk, I found he is not yet a tenured professor, but an associate professor. Very interesting system at Columbia University, I believe. I think it is a matter of time because he is already a big name in the field. Also, I thank Chong-Hae Wang, a graduate student at MIT, for his kind offer to give his 2022 talk spot to TL To, who originally wanted to speak today, but now he changed his plan in terms of job search. So he now will speak in 2022 instead of today. And this rearrangement allows Tal, today's speaker, to give his wonderful talk, I believe. And without, of course, further ado, Tal, the tall virtual podium is now all yours. Thank you so much for your time today.
Okay, all right. Thanks so much for that introduction, and also for fitting me into the seminar series for this year. I think it's a really nice series. So like Tae-Suk mentioned, my lab focuses on engineering bacteria for cancer therapy, and it's actually a really old concept. And I wanted to sort of start at the beginning, because it was over 150 years ago that it was serendipitously discovered that bacteria had an effect on the growth of tumors. And this happened when a patient came into the hospital, had a tumor on their neck, and got a bacterial infection. They actually were put in the bed of a previous patient that had a bacterial infection. This was before anything was known about sterility. And so they contracted this infection, but over the course of about a week or two, their tumor actually stopped growing. So this was the very first observation, and it wasn't until several decades later that William Colby, who worked here in the New York area, found this case and several dozens of other cases where there was a correlation between bacterial infection and tumor regression. So he really pioneered at a large scale this idea that you could try to use bacteria as a cancer therapy. And he first did this using live strains of bacteria, typically streptococci and serratia, and injecting them directly into tumors or into patients. And then he went on to use heat inactivated strains and concoctions of different strains that are known as Coley's toxins. And his methods actually met with significant success. So for certain kinds of tumors like bones and soft tissue sarcomas, more than 50, 60% overall response rate, and some cures were actually observed. Now, at that time, it wasn't quite understood how it was working. The host response was important, but really there was a poor understanding of the immune system. And later it was discovered that the bacteria stimulated the host immune system, and that was responsible for part of the therapeutic effect. For this work, he's known as the father of immunotherapy. But at that time, this is early 1900s. This is before the widespread availability of antibiotics, and also the beginnings of chemotherapy and radiation were becoming safer alternatives at that time. It really led to the demise of this line of research. So today, synthetic biologists like myself are starting to rethink this problem and trying to see how we could better engineer bacteria as a cancer therapy by improving their safety, as well as their efficacy. So one of the things that was not quite appreciated at Colby's time, and maybe partially responsible for some of the therapeutic effects that he observed, were that certain bacteria species have this unique ability to grow selectively inside of tumor environments. So this is a prototypical experiment that we do in the lab. We intravenously inject, for instance, an E. coli that's expressing bioluminescent genes into a mouse that has 2 hind-flank tumors. And what you can see is that quickly after 24 hours, you see a very strong signal inside of tumors and no signal in any of the other healthy organs or parts of the mouse. And what's happening here is that bacteria enter the tumors through the vasculature, and they enter healthy tissue through the vasculature when you initially inject them, but they're very efficiently cleared in healthy tissue because it's well fused. There's a lot of the components of the immune system present. But in tumors, there's a necrotic core, which bacteria essentially use as a safe haven to grow and hide from the immune system. And so they can actually grow inside of tumors, and they're cleared in healthy tissue. And that results in this biodistribution, if you count the bacteria, where there's high numbers of bacteria in the tumors, and none are present in the healthy organs, just 1 or a few days later. So this kind of approach is really interesting. It works for tumors that are about 1 millimeter cubed or larger. So it has to be post-angiogenic switch. As the tumor grows, it forms this necrotic core. It's not a targeting mechanism per se, so they're not targeting anything specific about the tumor, but rather they land in the healthy tissue, and they also land in the tumor tissue, and they only grow in the tumor tissue. And that's what leads to this very specific biodistribution. So many species of bacteria have been shown to grow inside of these solid tumors. They have to be able to survive the environment, which is usually hypoxic and low pH and has necrotic tissue. Salmonella typhimurium and E. coli are definitely the most widely used strains, and they're also the easiest to genetically manipulate. But other strains like Clostridium novae and Lactobacilli and Bifidobacterium strains have also been used and shown to colonize tumors. And so probably what I think is the most exciting aspect of this approach, especially in the landscape of cancer therapies, is that these bacteria have the ability to colonize essentially all solid tumor types. And they do so in a manner that they don't really care about the genetics of a tumor. And they do it in what we call an antigen-independent fashion. So rather than other cancer therapy approaches that target a certain pathway of a cancer cell or a certain receptor that may be specific to certain cancers, bacteria really can colonize a wide variety of solid tumors. And so in our lab and in other labs, we've demonstrated that they can colonize human tumors of various origins, mouse tumors, colorectal, pancreatic, ovarian, lung, both on flank models of mice, as well as in orthotopic models, as well as liver metastasis models and genetically engineered mouse models. And there's some data that I'm not going to spend too much time talking about today, showing some colonization in human studies as well. So the colonization aspect is very robust, and you may be wondering how exactly do they colonize tumors? And I would say it's been studied quite a bit, although it hasn't been completely elucidated. And I'm not going to go too deep into this today, but I just wanted to mention the highlights, which are that bacteria enter these tumors by mostly a passive process. So they enter through what is typically leaky vasculature that's not very well formed, and they accumulate by an EPR-like mechanism, sort of like particles. They don't need to actually swim or be motile to show the selective amplification mechanism, although it does, one group has shown that the motility can actually help enhance their distribution in the tumor. And then they cause a host response. And here you can see in these micrographs bacteria stained in green inside of a tumor. They're surrounded by different immune populations. And like I mentioned, these immune populations don't really go into the core. They can't clear this little bacterial colony, but they surround it actually and keep it quite contained. Now, while they're in the core, the bacteria also cause necrosis. So they compete with cancer cells for nutrients and for oxygen. And you can see here, they cause even more cancer cell death. They also stimulate anti-tumor immunity. They may be producing some toxins. And so just bacteria alone by themselves, without any engineering, could potentially have a therapeutic effect on certain kinds of cancers and for certain patients. So some examples of that are BCG therapy, which is an attenuated mycobacterial strain that has been clinically used now for many decades in the treatment and management of bladder cancer. So it's an attenuated bacteria. And a couple of other strains, more recently, this was in the early 2000s, were genetically modified, like a Salmonella typhimurium strain that is modified to change the LPS structure so that it reduces immunogenicity in patients, and you can inject it intravenously. And there's a couple of other mutations here. And in some of these early phase 1 clinical trials, it was demonstrated that bacteria could be injected quite safely at very high doses of up to about 1 billion bacteria, and actually showed some colonization in human tumors, but just the bacteria alone didn't really show any efficacy. And so today, there's a couple of ongoing trials, and really the field has shifted towards thinking the bacteria alone don't have a strong enough therapeutic effect. Let's try to engineer them to make payloads locally while still trying to maintain safety. So that's where a lot of the synthetic biology comes in. And in our lab thinks of bacteria as these intratumoral bioreactors that live inside the tumor and could locally produce and release therapeutics. And these could be protein therapeutics or peptides or even small molecules or RNA molecules or things like that. And I just mentioned this approach because there are several groups who are using bacterial therapies where a bacteria will invade a cell and deliver something intracellularly. Our group primarily thinks about bacteria as living extracellularly inside of the core of the tumor, and that mass acting as a bioreactor to locally release drugs in situ. And so the advantage there is that we can make high concentrations of a drug, even drugs that have been tested before in clinical trials, but have systemic side effects. So we can make them locally, avoiding some of the off-target effects that are observed. We've shown that we can kill them with antibiotics, and probably most exciting for this audience, we can engineer them to continuously sense and respond with synthetic biology approaches. And so some examples of that in the literature you can imagine are sensing A, B, and C about the environment and computing something and deciding on which type of therapeutic you should produce in response, producing all kinds of kill switches and containment systems that many groups have worked on, including Taesuk's group. And also in our lab, we think a little bit about how we can use communication systems, quorum sensing, et cetera, to have these bacteria not just act individually, but act as a community, which could potentially enhance their therapeutic effect. So, for this audience, I just want to give a little background on cancer immunotherapies. There's been, enormous amounts of success in the last decade or two, primarily through checkpoint inhibitors, which are antibodies that can enhance T cell activity, CAR T cell therapies that are engineered to sense and respond to specific antigens on tumor cells, and also oncolytic viruses, which are even more recently been developed that can replicate inside a tumor and also deliver some therapeutic responses. So even though there's been an enormous amount of success, there still are some really significant unmet needs, and those primarily exist for solid tumors, which are the majority of patients that are diagnosed with cancer today, and where the largest unmet needs are. And these primarily occur because of efficacy and safety issues with our current standard of care. And some tumors just do not have a lot of T cells that you could activate with checkpoint inhibitors. They have an immunosuppressive tumor microenvironment, so CAR T cells may not be active or replicating or be able to traffic there. And the solid tumor TME environment can also be a limiting factor for diffusion or for entry by viruses or even antibodies or CAR T cells. And then in addition to limitations in efficacy, there's also limitations in safety. So if you're targeting something specific on the surface of a cancer cell, that specific receptor might actually exist in other healthy tissue. And so there's what are called on-target, off-tumor types of toxicities with many of these approaches, as well as just off-target effects. Having many of these therapies, as well as combinations of these therapies, can lead to what are called immune-related adverse effects. So we think in our lab that bacteria have some unique properties, particularly because they can target these solid tumors so well, that they may actually have advantages as compared to some of these other therapeutic platforms. So I talked a little bit about synthetic biology and these therapeutic platforms, and I'm not going to spend too much time on this today, but there's also a lot of evidence that there are not only microbes inside of all the healthy tissues inside in your body, but also inside of patient tumors. And it varies between tumor, and it depends on the type of tumors and their location and things like that. But a lot of people are now thinking, well, if bacteria are already there in patients and not causing recurrent infections, that maybe we could even use those bacteria that are present or manipulate the ones in situ to naturally act as a platform for drug delivery. So in our lab, we think about engineering bacteria in 4 different axes. One is the strain, what kind of strain or species should we use? Another is the payload. Another one is the kinds of genetic circuits that control safety or efficacy, control the bacterial behavior, and also their clinical use, meaning what types of cancers and delivery route, et cetera. So when we started the lab, we really started focusing on what kinds of payloads would be interesting with bacteria. And we had a genetic circuit that was already developed, and just try to think about the coupling between these 2 axes. So we used what we called the synchronized lysis circuit, which was originally developed in Jeff Hasty's lab by Omar Din. And we worked on some of the therapeutic aspects of this circuit. And it consists of a positive feedback quorum sensing system, whereby when bacteria grow and they hit a critical threshold, they then produce a second phage lysis gene, which will cause them to lyse and burst open. And the idea is that you can release a recombinant payload effectively without having to deal with secretion tags. Now, interestingly, not all the bacteria lyse when they hit a quorum. Actually, a few bacteria survive, and then they grow up again and reach a critical density and could cyclically produce these lysis events. And you can see that here in this movie. So this is a microfluidic device with bacteria growing. You can see that they produce GFP here as a proxy for the therapeutic. They grow up, they lyse, and they actually leave behind what's called the ghost of bacteria, the remnant membrane. So think of them in the tumor as being able to cyclically deliver a drug on a particular dosage, with a particular dosage and frequency that you could genetically tune. And not only releasing the therapeutic, but also releasing all of their intracellular contents and the LPS that's in the membrane to intratumorally stimulate the immune system inside of tumors. There's some other interesting advantages where this could act as a safety mechanism, and the quorum sensing acts as a specific tumor signal. So we started investigating this therapeutically. So Shreyan, who was a graduate student in our lab, started looking at the SERP alpha CD47 axis, which is known as an innate immune checkpoint. So CD47 is expressed on every cell in your body, healthy cells, as well as tumor cells. And it's used as a don't eat me signal to so that phagocytes in your body could recognize your cells as self and not just eat every cell. And now tumor cells express CD47. And then, in fact, they express it at both high frequencies and also high fold change compared to normal tissue. So it's in some ways a strategy for tumor cells to evade getting eaten by phagocytic cells like macrophages, which would then present these tumors and fragments of the tumors to the immune system. So there's many groups who are trying to block this interaction. So it would lead to increased phagocytosis, and antibodies are the canonical way to do this. Those are quite challenging for us to produce in bacteria. And so we focused on nanobodies, which are derived from these single heavy chain fragments of for single heavy chain antibodies of the camelid family, like llamas and alpacas. And they have these really interesting VHH domains that have very high affinity for their target in the picomolar range. But they're also a small size so that they can diffuse well, and they have short half-lives, meaning if you try to inject them systemically, as compared to antibodies, they don't accumulate in tumors that well. And so for us, that was an interesting coupling because we can make a high concentration of these locally inside of the tumor that could then have a therapeutic effect potentially. So we expressed these in our lysing bacteria and injected them into A20 lymphoma tumor model. And we found that we could completely clear these tumors over time with bacteria expressing the CD47 nanobody. In this model, bacteria alone have a therapeutic effect. You can see that eventually the tumor starts to grow back after you stop injecting bacteria, in this case in an intratumoral dosing regimen for the first 11 days. So that was really interesting to us. And we went on to do this really neat experiment where we now had 2 tumors, but we just injected a single tumor, the left tumor here of this mouse, and we found that 4 out of the 5 mice had their tumors clear, but now we also saw a response in the untreated tumor as well, and no response from just the bacteria alone. So we went on to investigate this further. All of this work is done very closely with Nick Arpaia's lab, who focuses a lot on the immunological aspects. And we co-advised Shreyan together. And what we found that it wasn't actually the bacteria that trafficked from one tumor to the other. You can see here, if we count the bacteria in the treated tumor, they stay in the treated tumor. None are found in the untreated tumor. Eventually, by day 30, the tumor has essentially cleared, so there's no bacteria in there. And there's also no bacteria in the liver, the spleen. So it wasn't the bacteria migrating, and we also had evidence that it wasn't the therapeutic getting out and migrating to the other tumor, but rather it was the bacteria stimulating anti-tumor immunity in this tumor that then those cell populations migrated to the other tumor. And so in the untreated tumor, we found that there was increased activation and proliferation of CD8 positive T cells, as well as interferon gamma production. And we did this really neat experiment where we actually harvested the tumor, homogenized it, and re-stimulated it with a very specific peptide, which is an MHC class 1 restricted peptide that corresponds to the unique phenotype expressed by the A20 lymphoma. And so it's a very specific peptide, and there we saw a response specifically in interferon gamma production only in the CD8 T cell population, as you would expect. So we're getting a very antigen-specific response. We did a lot of controls that were part of this paper. We showed that the bacteria alone had some therapeutic effect. The antibody, not the nanobody, the antibody had some effect. Co-injection of those two has some effect. The lysis has some effect. The recombinant antibody has some effect. The bacteria without the lysis circuit have some effect, but you only see this complete clearance when you have these live and lysing bacteria that are able to stimulate anti-tumor immunity and produce this durable and long-lasting therapeutic response. So that's the mechanism that we worked out so far in this system. It works by 2 modes. I didn't quite mention this, but one is the nanobody blocks the interaction of CD47 SIRP alpha, which leads to increased phagocytosis. Then the lysis of the bacteria has an adjuvant property to also increase phagocytosis, which we think broadens antigen presentation and leads to a tumor antigen specific T cell response, and then clearance and systemic anti-tumor immunity. Okay, so this was a proof of principle for us. Candice in our lab then went on to improve technologically the system. She genomically integrated the synchronized lysis circuit into E. coli nissl, expressed some other nanobodies, PD-L1 and CTLA-4, which are checkpoint inhibitor nanobodies, and showed now with just a single injection, we can clear tumors and actually outperform the systemically delivered antibodies that are used today in the clinic. These are the mouse analogs of those. In other tumor models, like colorectal tumor models, where there's not they're called colder models, there's not a lot of T cells present, we don't see as strong of a response with the checkpoint inhibitor nanobodies, but if you add a cytokine like GM-CSF to recruit T cells, we can actually then improve the therapeutic response. So we're really interested in finding these ideal combinations of payloads with our bacterial strains. Now, I started off talking about systemic delivery, and I think that's really the end goal. So in the case of intravenous delivery, we could see here that bacteria improve tolerability in terms of, let's say, they reduced the numbers of platelet counts comparing to an antibody. So this antibody would be blocking CD47 on healthy red blood cells and platelets and then lead to phagocytosis. But in the case of the bacteria, we don't see a reduction in platelet counts. We also don't see a reduction in body weight, and we have this nice biodistribution. So that all works quite well, but it turns out what we found is that intratumoral delivery always has higher efficacy compared to intravenous delivery. These would be the comparisons for the CD47 nanobody, and these would be the comparisons for the PD-L1 and CTLA-4 nanobodies. And so that was sort of a mystery to us, and it still is, because we thought if you just get a couple of bacteria in the tumor, within 24 to 48 hours, when we look, there's essentially the same numbers of bacteria between intratumoral and intravenous injection. So why does that early injection of bacteria and that short time scale really matter so much? We don't completely understand that yet, but what we did understand is that the more bacteria you can inject in the tumor initially, the higher the therapeutic response. And so we developed a strategy based on this, where we induce these capsule polysaccharides on the surface of bacteria, and that these could potentially modulate the immune evasion and the clearance of bacteria. And so I think naively the idea is, well, if you coat bacteria, it could be a chemical coating even, that will allow them to be protected and potentially have higher circulation and lead to more bacteria in the tumor for the same injected dose. And what we found is that that's true, but it also leads to more toxicity and slower clearance of the bacteria because of the slow clearance. And if you remove this capsule altogether, that actually has high immunogenicity because you're exposing LPS and other factors. And so that has initial transiently high immunogenicity, but then it actually decays quickly because these bacteria are cleared pretty rapidly. So to try to get the best of both worlds, we built a system where we can inducibly control these capsule polysaccharides and have them induce it initially and then have them turn off as they get to the tumor or as they're circulating in the bloodstream. And it turns out that can actually shift the maximum tolerated dose of our ICAP system as compared to the wild type E. coli Nissl that have these capsule polysaccharides, or even a knockout strain by about 10 to 20 fold. So this shifts this therapeutic window, which means you can inject more bacteria for the same amount of toxicity and get a better and improved therapeutic response. We had a couple of different models with bacteria expressing a toxin that has a therapeutic effect. So this is really interesting, and we found that one of the things that you could also do with the ICAP system is induce the production of these capsule polysaccharides in situ, inside of a mouse, inside of a tumor. And so here's a mouse that didn't have the capsule polysaccharides, it's inside of the tumor, then we delivered IPTG, and then these bacteria migrate to the other tumors inside of the mouse. So we can inducibly have them migrate to other tumors or potentially metastases, or have them circulate in the bloodstream to search for other tumors that are present. Okay, so I don't know how much time I have left, but I'm going to go quickly through just two last examples. The first one is, I think many people have thought about this idea to engineer biosensors for a specific niche. For us, it would be the tumor, which has certain hallmarks like low oxygen, high lactate, and somewhat lower pH. In our hands, we struggled with this at first because these biosensors do not have a very high fold change with respect to the tumor and normal conditions, maybe sometimes 4 to 8 fold or so. And so the strategy that we enacted was to have bacteria not drive for instance, a therapeutic with these poorly performing biosensors, but drive growth. And so we would knock out an essential gene and produce the essential gene under these promoters. And in fact, we can actually combine multiple promoters together. So the one example I'll share here is one with lactate and hypoxia, driving 2 essential genes, ASD and GLMS. And you can see that these bacteria only grow in the presence of lactate and hypoxia. They don't grow in either of the single conditions. And what that does in terms of escapee rates is improve the performance. Now it's essentially a multiplication of these independent probabilities of escape that come from the single sensors. And so our escapee rates are typically around 10 to the -8 and are improved by this multiplexing and essential gene strategy. And in the tumor context, we showed in a mouse model that after IV injection, the hypoxia lactate circuit could still grow to the same levels in tumors and not grow inside of the liver, where there is some hypoxia present and some levels of lactate. And what that does in terms of tumor to liver ratio, which is how we think about the specificity, is improve it about 10-fold or so. So we can actually get some levels of improvement in terms of specificity. Okay, so now just zooming out broadly, I think at the very beginning of the talk, I mentioned that Colby found that certain kinds of bacteria like streptococci worked very well for certain kinds of bone and soft tissue tumors. And so the limitation for us has always been the animal models and testing them. You can't test that many therapeutics in a high throughput fashion in animal models. So Tetsu in our lab has been working on this bacteriospheroid platform where we can make these cell lines into these spheroid aggregates and then add bacteria to the surrounding media along with some gentamicin. In this case, you'll see some salmonella expressing GFP. And what they'll do is invade these tumor spheroids and grow in the hypoxic and necrotic core that develops inside of the tumor spheroid. And this resembles a 3D environment with hypoxic and other similar conditions to the in vivo environment that we've looked at. If you don't add antibiotics, they just grow everywhere. So now we can do some interesting imaging types of approaches, look at the concentrations of bacteria over time for usually about the course of maybe 1 to 2 weeks or so. And what that allows us to do is monitor gene circuit expression in these 3D multicellular systems with different kinds of circuits. We can build different libraries of therapeutics. These are a collection of a couple of toxins and anti-cancer peptides, which can inhibit the growth of spheroids. And some candidates here stick out a little bit more than others. And probably what you can never do inside of a mouse is now test all the different circuits against all the different therapies. And so this was really interesting to us because you can see for some therapies, if you look at quorum sensing, first quorum sensing with lysis, some are improved by lysis, meaning they need to actively be released, and other therapeutics have the ability to actually release themselves. And that's been known in some cases, but this allows us to try to develop strategies for bacterial cancer therapies. Okay, so now zooming out even further, I think I mentioned that there are all these other technologies in the cancer immunotherapy landscape, and we're really interested in comparing to these technologies and also combining our bacteria therapy approach with many of the technologies that you see here. And so to that end, we've recently created this probiotically guided CAR T-cell system that we call Procars, where bacteria colonize tumors and they can release molecules that act as beacons for these engineered CAR T-cells. And so one of the limitations of the CAR T-cell approach is finding antigens on the surface of cancer cells that are safe and specific, that don't exist anywhere else in the body and are not exactly heterogeneous. And tumor cells can actually have resistance to these by just removing the antigen expression altogether. So this is an approach that we developed where these bacteria could actually release these synthetic antigens. And then these engineered CAR T cells are then able to recognize these synthetic antigens and enact killing response on the neighboring tumor cells. And we've shown in vitro that leads to specific lysis. And in vivo, Rosa and Candice have shown that we can actually get improved therapeutic response in that triple negative model by combining the bacteria and CAR T cells together. Okay, so I hope I got a chance to tell you a little bit about bacteria and the different ways that we're engineering the therapeutic payloads, the safety switches, the different cancer delivery routes and combinations with other therapies. And I want to just end on this one last note, which is that our lab is also really excited in sharing the vision of our work through creating artworks with cancer cells and with bacteria. And so this is a piece I created with Vic Muniz, which is actually a mandala that is made entirely out of cancer cells. You can see them here as we zoom in. So this is one of the things that we believe in, which is that visual art allows us to communicate science in this very universal way. It's been very important, and especially research times. And we get to connect a little bit closer with the human experience, particularly for cancer patients. And that's one of the reasons we try to engage at a fundamentally deeper level with the work that we do, and try to inspire some new and creative innovations for the future of cancer research. So with that, I want to thank everybody in our lab and our funding sources, and again, Taesuk and the organizers for inviting me to speak to you here today.
Wow, wow, wow. Amazing work and talk. I love it. Of course, I mean, this is a very important topic, given my personal experience, including my wife had 2 benign tumor surgeries. And also, I believe many of you might have some family members suffering from cancers, as humans are living longer nowadays. And TAL's approach is really innovative and critical for our society. So thank you so much for your wonderful work. Also, I believe climate crisis, as Lars mentioned, would lead to catastrophic consequence, potentially killing millions of people, probably even more people's death than the COVID killed for the last 2 years if we do not act right now. So we should address this issue right now. So of course, I mean, despite all gloomy current situation, I'm very optimistic because we have fantastic scientists and engineers like Tal and Lars, also young people working hard every day to solve many problems we have. So we have already tons of questions. I want to ask Lars, speak up by unmuting first, and then I'll address all other questions we already have.
Okay, Tal, thank you so much for the really inspiring talk. You very nicely showed the 3 topics. I'm obviously interested on your choice of microbe, and obviously you couldn't put more into your talk, but just give a flavor. It's the microbe, the host, has to be non-immunogenic, and you told us via the capsule, then it has to be still protected. And what was a polysaccharide you used? So if you give just a little bit of flavor, what is really the importance on the host?
Yeah, it's a really good question, and I think it was just due to time. But the bacterial strains and species is kind of like the next frontier, I believe, for this field. I think there's always going to be the synthetic biology, and people are going to stick to E. coli and Salmonella probably for that. But I think what are the ideal properties of a host that you want in this situation? It's a little bit of a double-edged sword, because you want them to be intratumorally immunogenic and immunostimulatory. So in some ways, things like LPS and flagella, which stimulate TLRs and other things, they have been shown to turn macrophage polarization and things like that, are important. But then outside, when you deliver them systemically, you don't want them to have those properties. So that's why I think that the switching mechanism is kind of novel. It's almost like two different species of bacteria that you want to switch between. But I think that there's so many interesting, generally regarded as safe bacteria that you could potentially use. So you could go the opposite way, which for us is we start with gram-negative bacteria. They're generally immunogenic, and we try to make them safer and control them. But you could do the opposite thing, which is to start with some probiotic lactobacilli and potentially make them, when they get into the tumor, more immunogenic or more immunostimulatory. So I think there's a lot of strategies that you could do there. And just even besides that, thinking just broadly, maybe there is already out there some amazing strains of bacteria already with those properties. We should be searching for them. And I think we're trying to get at that with some of the spheroid assays and TLR-based assays to kind of do some bioprospecting like that.
That's fantastic. And also your photos, your microscope. I think this communication is really an important part. Yeah, worked for me as well. So thank you.
Thank you so much
Thank you.
for a wonderful question and answer. So we have lots of questions. So I will start with the Joe Min's question. So great talk, thanks. Regarding the nanobody treatment antibody side, what exact epitope for targeting VHH, the CD47 tumor side, do you think tumor will develop some way escape from this antibody blocking the interaction between CD47 and SIRP?
It's a very good question. I realized that I should have said this. I just forgot to. There was a reference on the nanobody slide, which is that we used a previously developed nanobody from Chris Garcia's group in collaboration with Hida Plua's lab. So they had already inoculated an alpaca and developed this nanobody sequence that they showed binds to mouse and blocks mouse CD47 SIRP alpha interaction. So we just adapted that sequence and expressed it in the bacteria. So I just want to make that clear. So they did all of the binding assays, and the epitopes are fairly well defined. It's the same epitope as the MyHap301 antibody. So it's a blocking nanobody. I don't know the specific protein residues or anything like that. I'm sure you can look it up. But I think the second part of the question is also really important, which is what are the escape mechanisms? So I think in this case, the good thing is if CD47 were to be downregulated and tumors stop expressing this antigen, that would not be a bad thing, because then you would also have increased phagocytosis just naturally. But there are other escape mechanisms that I could think of, which are, there have been people in my lab actually that are interested in looking at whether nanobodies that we produce in bacteria could develop an antibody response from the host. So now you make a lot of these nanobodies, maybe the host starts blocking the nanobodies with antibodies. I don't think that's the case, because I don't think antibodies diffuse that well into the solid tumor environment. There's very poor diffusion. And so things like complement do not really get in there to clear. So I think the numbers will work in our favor, but I think it's something that we definitely want to look at and think. Probably the future question is like, okay, so we did this treatment, it kind of works, but what are the resistance mechanisms, and what are the next level of treatments that we can make to kind of improve the resistance from the host or even from the bacteria?
Yeah, so challenging problem, I guess. So, but I believe you will solve the problem future, of course. Okay, so another
Hopefully,
question is another question from Matt. How many survivors of the SLC are escape mutant and give rise to non-functional daughter cell? And how many are random chance survivors with fully functional progeny?
Yeah, a very good question. So the longest that we've looked so far in the recent 2020 paper was, we found that after 2 weeks, the bacteria synchronized lysis circuit with the synchronized lysis circuit integrated into the genome are still lysing inside of mouse tumors. So that's the longest that we've looked. And we then homogenized the tissue and plated single colonies and looked for whether these colonies still had the behavior to lyse. And essentially 100% of them do, maybe 99%. So we think that it's quite functional at 2 weeks. That's sort of how we picture the cancer therapies. So, I mean, the dream would be these bacteria are living in the body or in a tumor for years and monitoring for resistance, right? But in practice, in some clinical trials are doing this now, is really something maybe more practical, which is to inject bacteria, and then maybe 2 weeks later, you could inject more bacteria. You could even clear out the previous bacteria with antibiotics, and it will be like chemotherapy and like other therapies, a multi-dose regimen that patients experience. So we don't have to get like a perfectly robust system to evolution, which is one of the biggest things that we're fighting. But I think it's a really important question. And I probably, in the cases that we're seeing with intratumoral delivery, we inject so many bacteria, they're almost, near the, I won't say they're near the carrying capacity, but they're closer to the carrying capacity than intravenous delivery, which just starts with fewer bacteria. So there's less of a chance for them to mutate in that course of a few weeks as compared to maybe if you just started with one bacteria in the tumor that grows and then could actually escape. So I think it's a very important problem. But hopefully in this first iteration, we can kind of get at something a little practical.
Okay, so probably the last question, but a little bit longer question. So thanks so much from Ahmed. Thanks so much for sharing your amazing talk. My mind is blown with regards to the cell lysis strategy to cyclically, release the therapeutic. Why not use column sensing to attenuate population growth and switch to therapy synthesis and secretion, including exporter synthesis? Interesting. This may encourage more continuous treatment at higher dose and help evade host immune response that you showed could clear the therapeutic bacteria. And just to be clear, I mean, but why not continuous production of therapeutic at low growth rate instead of cyclic release of therapy by lysing?
Mm-hmm. Yeah, I think it's a really good question. So I think there are a couple of advantages. There's obviously pros and cons. So you could use things like type 3 secretion, and there are groups and companies that are injecting, using bacteria to inject proteins directly into cells. But maybe you're also referring to how would we compare it to a secretion system just into the extracellular medium? So in our experience, and I could say this pretty generally, export sequences and exporting things out of bacteria can be highly strain and protein specific. There are limitations in terms of the amount that you can get out, the types of sequences, the sizes, and things like that. There's also a little bit of immunogenicity associated with those complexes inside of bacteria. That may not be a bad thing. So I think for us, we have tried a couple, and it takes a lot of tweaking. And I don't think it's a bad idea. So nobody, to my knowledge, has compared like a secretion strategy to a lysis strategy. I think that would be a super interesting study. And I think that the point that the question was making was, if you deliver things continuously, perhaps that's better. But it's actually also true in the cancer therapy literature that there's this whole idea of doing something called metronomic therapy, which is not delivering at a really high dose, maximum tolerated dose, but delivering at smaller intervals with a higher frequency. And that's been shown to actually alleviate some side effects, actually some cases increase efficacy, and in some cases decrease chemo resistance. The last one you can imagine, because if you don't just give drug all the time, there's potentially less of the ability of the cells to develop resistance if they only see it for a short period of time. So in some ways, when you have this lysis strategy, you kind of build up the drug and then you release it all at once. You can release more than a typical like EC50 threshold like that you might see with secretion, with like kind of a slow drip. You can release it all at once and then kind of let it kind of grow back in, and then do the same thing over again. So we haven't shown that that's the case. I think there are cases where one would win over the other. And I think the last point that they made was also good, which is if you lyse the bacteria, they could actually get cleared at the lowest point of their population, more so than a growing population. So we don't know the answer to that, but I would say that the lysis circuit is also interesting because it could actually act as a safety mechanism. It grows to a certain concentration and then lyses, and it never grows too much before it lyses again. So it has like some advantages in terms of safety and like overgrowth compared to like a secretion strategy.
Yeah, I think that's kind of right approach, or the good approach, I believe, because I mean I also know some other case. I mean one of the first synthetic biology probiotic paper about killing pathogen, but that also used the lysis done by Matt Chang, and actually that paper scooped the Ron Weiss approach. I mean they wanted to secrete the protein out, the peptide out. So and then also secretion is very, difficult, in a sense. I mean, I guess, plastic case, we probably want to secrete the, plastic degrading enzyme out of the cell, but that was a very challenging problem. In that you could do the consolidate process, kind of using, not rather than using two-step pathway, degrading first and then, utilizing monomer, but we could do everything one step, but secretion is kind of key, kind of challenging, step to do that. So I think I need to stop. So because it's already, the 11:04 my time. So thank all for joining and staying today again. We'll meet again next week on December 9, Thursday, the same time. We'll have Professor Sarah O'Connor from Department of Natural Product Biosynthesis, in fact, from also Germany again, and also Lennart from Leiden University, Netherlands. So both from Europe again, although Sarah actually used to be at MIT in US. And as usual, the following follow-up in the Zoom chat will occur via the link in the chat box I posted. So see you soon. And I hope Tal, stay a little bit longer in that another chat. So you could click that on and then you just say yes to transfer. You will automatically fly over to another room.
Okay, sounds great.
Thank you so much, Lars. It's a great honor to have you today. Okay, so meet you there, in a minute.